Thank you, Rebecca. And thank you for the invitation to give a little talk here about a few viruses that are serious issues in cucurbits. The first one that I'll be talking about today is cucurbit aphid-borne yellows virus. So here's a few pictures showing the main symptoms associated with this virus and a couple of the key vectors. It initially appeared in the United States around the 1990s. After shortly after being discovered for the first time in France. And it was described around the same time that it appeared. The first location where it appeared was in California, in the Sacramento, San Joaquin, and Salinas valleys. And it was widespread in a variety of different cucurbit crops. So surveys at that time indicated that it may have been here for quite a while and was already well established. And actually, in my own research, I have found that it's also widespread in wild cucurbits that are very common in the Southwest. And so this may have been the reservoir that held this virus for some time before it showed up in crops. It can also infect beet, lettuce, and a number of common weeds. And the transmission is mainly by the melon aphid, and that's shown down here in this first picture. So where is this virus in the United States? You may hear it referred to as CABYV, as CABYV or CABYV by the people who study and try to prevent infections by this virus. I will try to stick with CABYV for clarity's sake. We often abbreviate our viruses. So these stars here on the map show where the virus has been detected in cucurbit crops. But you'll see that I've placed a number of different markers on here for areas where it's likely present, but hasn't been rigorously sampled for yet. The virus is distributed broadly throughout the world in areas that have sort of an arid Mediterranean type climate or a subtropical climate. So you can see that in the map up in the corner here. So any cucurbits that are growing in an area with that type of climate that has the vectors present could potentially be a place where this virus could be present and causing disease. So we looked at a couple of pictures of the symptoms associated with this virus in the initial picture. Here's a few more showing the range of ways that this virus can manifest in the plant. And thank you, Bill Wintermantle, for these lovely pictures. So the main symptoms, if we could list them, this is what we would expect to find. Is this yellowing of the leaves, and especially on the older leaves, right? So these are the leaves that are closer to the base of the plant rather than the newer leaves that are at the growing tips. You might see this characteristic vein banding, where you see that the veins are darker than the leaf tissue in between. And then down here, this picture shows that leaf thickening and brittleness that occurs in some crops. In terms of yield impacts, the main issue is that there is flower abortion caused by the virus. So the plants are either producing fewer flowers or they're aborting the flowers before they can be pollinated. And so where there are disease impacts on yield, that's where we would expect to see it. But of course, these are just a few pictures of typical symptoms. And you're going to have a lot of variation depending on which crop is being grown, which virus genotype or isolate is in the area, and when the plants become infected and their growth stage. So you should be aware of that when you're trying to decide whether this virus might be present. And of course, just like all virus issues, it's very difficult to diagnose infections and ascribe them to a particular virus just based on symptoms. So there's a lot of physiological disorders, including nutrient deficiencies, and even drift from herbicides and other chemicals that can induce symptoms that are very similar to viruses in the plants. And of course, many viruses cause symptoms that are very similar to each other. All right, so if we had to ask, are these both pictures showing infections by CABYV? They look almost identical in terms of their symptoms, but these are two very different viruses. The one on the right is a whitefly transmitted virus that was talked about in the previous webinar. But their symptoms are almost identical in cucurbits. So we can't use symptoms to diagnose CABYV infections. If you want to know that this is the virus causing disease in your crop, you would have to use a molecular diagnostic or a serological diagnostic. Serology diagnostics use proteins in the virus, and we use a test binding something to that protein that lights up and is visualized. And then we can tell that it's present, and it's very specific for the proteins only in that virus. And these other ones listed here act by amplifying the genome of the virus many times so that we can see that it's there, right? And this last one right here, high throughput sequencing, very expensive but untargeted approach. So you put all of the genetic material through this process, and it sequences and it will tell you all the viruses that may be present in your plant, maybe more than just the one causing disease. But we don't have access to these typically as growers or PCAs, and even a lot of researchers don't even have this capability in their labs. So it's important that you look up your local plant diagnostic laboratory and inquire with them about testing capabilities. And you also should seek out some advice from your extension agent because they might be able to point you to a laboratory that could provide this service for you. Thinking more about how this thing is spreading in the landscape and why it's getting into your crop in the first place. The main vector, that is the insect that is moving this thing around, is the melon aphid, but it can also be transmitted by the green peach aphid and the potato aphid, although those are typically not as big of an issue in cucurbit crops, and they're very poor efficiency with transmitting this virus. So this is the main insect that we're concerned about. So scouting for that insect, looking for that insect may give you a clue as to whether this virus could be moving in your crop. How does the transmission occur? For CABYV, the virus particles that the insect has to pick up, they are inside the vascular tissue of the plant. So that's called the phloem, and it moves around all the nutrients and sugars in the plant to all the different parts, right? So this is the part where the insect wants to feed so it can get all that nutrients for itself. And to acquire the virus from this location, the aphids have to get in there and spend a fair amount of time feeding to get enough virus in their body to be able to transmit. Once they take the virus into their body along with that plant sap, it can move actually through their gut wall and enter their salivary glands, where it can sit there and wait for it to feed on another plant. When the insect does that, it will inject saliva into that plant as part of the normal process of feeding. And if the conditions are right, the virus will infect that plant and replicate and spread and potentially cause disease. So thinking with that in mind, we can think about some of the epidemiological factors that play into the spread of this virus, the transmission mechanism of this virus, and how it becomes a problem in a crop, and some of the management tactics we might implement to address these epidemiological factors. So the important thing to remember is that typically in cucurbits, we're dealing with mixed infections. So we might see infection by CABYV with mosaic viruses or with other yellowing viruses, maybe transmitted by whiteflies. So it becomes really difficult to actually understand how much of a problem this virus is when it's often co-occurring with many other viruses. And to be honest, the impact of this virus is really not well quantified. The main place where it's a problem, if it is going to be causing disease, is in the Southwest, where it's more widespread. And the most vulnerable period would be late summer to fall, when we have those amplified vector populations due to lots of other things for them to eat growing in the environment. So you can adjust planting times to avoid exposing plants at the earliest stages of growth to these higher vector populations later in the season. And you can scout for those vector populations and be aware of what those numbers are prior to planting if possible. The virus and the vector also use common weeds as alternative hosts. And so controlling weeds and especially volunteer cucurbits near fields is very important for managing incursions of viruses into the field. So if you can keep it from getting in there in the first place, you might be able to prevent that outbreak. And an important thing about the transmission mechanism is that once that aphid vector picks up the virus, it's pretty much going to be able to transmit it for the rest of its life, right? So it can hit many, many plants and potentially inoculate lots of plants just from a single vector. So when you keep that in mind, with this kind of virus that takes this kind of transmission mechanism to spread, you can use systemic insecticides to prevent the insect from reaching the phloem and thus prevent that acquisition or inoculation. There's a couple that I've listed here where we have some evidence from lab testing of reductions in the number of aphids reaching the phloem and the time that it takes them to get there. The best idea would be to consult your local extension agent about the possibility of using some of these in your management practices. There's also variation in cultivar susceptibility, but this is not rigorously quantified for this virus. There's a couple of genes that have been identified to confer partial resistance and tolerance, but they're not widely available yet. So that's unfortunate, but there may also be variation in cultivar sensitivity that can be leveraged even if we don't have a genetic mechanism identified for that yet. So talk to your extension agent and also consult university websites that list typical resistance traits in common cultivars that you might be using. And with that, I will end my presentation on this virus, and I will turn it back over to Rebecca.
All right, thank you, Kerry.